Imagine a flock of birds flying or a school of fishes swimming in a coordinated manner, and now shrink this idea down, way down to a cellular scale. Hello, I am Rohit Nautiyal, a second-year PhD student working in the lab of Xavier Tripat. Cells in tissues can self-organize in a similar way, giving rise to a collective phenomenon which is important in many biological processes that requires large forces. In tissues where cells are elongated, something really cool happens. In these tissues, cells align to their neighbors, forming big patches having same orientation. And between the patches with different orientations, we find defects, and these defects can generate very consistent pattern of forces. One of the tissues in the body that presents alignment are found in the interior of blood vessels, which we call endothelia. But how the cell alignment and defects influences the behavior of endothelia has not been well understood. In particular, how these defects control the forces within endothelia is a topic of interest. And to understand these forces, I use a technique employed in my laboratory, which is called traction force microscopy. In this technique, what I do, I grow cells on a soft and flexible surfaces which are coated with tiny beads, and when these cells exert forces, these tiny beads move. So by tracking the movement of these beads, we can actually study how the cells generate forces and organize within the tissues, especially around these defects. Further, I would also like to explore how these aligned and defect regions within the endothelia are controlled by controls or response to deformations like controlled stretching and compression.